Kristala Prather is the next speaker. She's Arthur Little Professor of Chemical Engineering at MIT. The title of the talk is Building Microbial and Chemical Factories.
Okay, so it's very nice to be here. I realize all of my friends from out of town probably had a much easier time getting here this morning than I did coming up the Southeast Expressway. But what a wonderful day to talk about climate change. I'm going to try to give, I recognize it's a very academically diverse audience, an intellectually diverse audience, and so I'm going to try to give more of a higher level overview of the kind of work that we do in my lab. With some examples, and then try to put it into context for the topic of this workshop. And I think Chris and George both alluded to some things that I'll touch on as well. The title of my talk is Building Microbial Chemical Factories. I am a chemical engineer, and the motivation for the work that we do, I'll be honest, was not originally driven by greenhouse gas reductions. I've been at MIT for 15 years. And I got interested in this idea of how do we think about transforming the chemical production industry, going away from hydrocarbons. I borrowed this phrase from a colleague of mine, Brad Olson, who says we need a paradigm shift from being dependent upon hydrocarbons to carbohydrates. And the way that we've traditionally thought about this is that if you look at right now what the current status quo is as far as where most of our stuff comes from, that's the best way I can think of to describe it. It is based on fossil inputs. We all know that there are tremendous environmental impacts and climate change impacts associated with that. And originally, or I should say early on in my career at MIT, there was a very big push towards biofuels, and the focus was really on the fact that so much of our renewable energy and so much of our fuels were coming from petroleum. What's also true, and I think that we're acutely aware of this as chemical engineers, is that there's a lot of materials that come from fossil fuels as well. And so one of the ways that I like to describe this is that we can think about electrification of the vehicle fleet and say, fine, we don't need any oil at all in order to have transportation. So imagine we get that for a future. But we're all, well, I'm standing, but you guys are all sitting on chairs or covered with fabric, and there's a really good chance that that fabric is more than 90% oil-based, right? And so we can't look at solar as a way of replacing stuff, right? We can look at as chemical engineer, we think about mass balances, energy balances. There's an energy component of this, but there's a mass component to this that we have to be cognizant of as well, which is where we're actually going to get the physical materials that allow us to do all the things that we do, and that includes being able to produce medicines, be able to produce materials, and so on and so forth. So we've been focused in my group on this idea of being able to now use renewably sourced inputs to be able to produce the stuff that we need. And so again, biofuels is where a lot of this focus has been. We've done some work on biofuels. I'll give you one slide more as an example of a kind of work that we do, but we've been much more interested in how do we think about the material side of it, where again, you can't think of replacing that with alternative forms of energy. So when I talk about microbial chemical factories, what we're talking about is the fact that biological systems, microorganisms in particular in our case, are actually just a collection of a lot of chemical reactions. And so this is a representation of cellular metabolism from something called KEGG, where each of the dots on here represent a specific metabolite. So that's a chemical. And the lines connecting those dots represent transformations. That is, chemical transformations that are mediated by enzyme systems. And what we've been focused on is how do you start with a very simple input, something like glucose, and navigate this very complex network, keeping in mind that there are a number of byproducts that are produced, and the cells also need to be able to take that substrate to grow. So what we've been focusing on in the group is really two sides of this issue. One is how do we actually increase the capacity of biological systems to do chemical synthesis, and I describe that as increasing the number of nodes or the number of points within that diagram that I showed. And then how do we actually increase the ability to do this productively? Productively in our case means at levels that are actually going to make an impact. For the context of this workshop, you can think of it in terms of having a significant impact on reducing greenhouse gas emissions. So I'm a chemical engineer, and I actually teach our introduction to chemical engineering class, and this is a classic diagram from that class, which basically says you have these systems, and there's inputs, and there's outputs. So if we think about that in the context of this problem, there's a couple of points I want to make here. One is to ask what can we make compared to what we need to be able to make if we think about using biology to be able to get access to the stuff that will allow us to remove our reliance upon fossil-derived inputs. What are the feedstocks that are viable compared to the resources that we have available? So George made the point in the sense of looking at various biospheres about the ability to be able to recycle. So if we want to think about removing greenhouse gases, but then also not making sure that we're continually pumping them back out, how do we actually have things be as recyclable as possible? And then within that control volume, there's a question of what's our flexibility? What does synthetic biology give us in the context of the ability to really be able to make larger, more complex changes and more complex systems to be able to push this forward as much as possible. So I want to start by talking about the things that we can make. And I'm going to talk about a couple of older examples in my group first, just in terms of how we've thought about this problem. And here's an example here, a molecule called hydroxybutyrolactone. There's a bunch of words. The point here is to say that when I first started the group, we were interested in, can we actually get biological systems to be able to make compounds where we don't already know what those biological pathways are. So if you think back to that image of all the dots, if I know what those dots are and I know what those lines are, I can pull them from one organism and put them into another organism. Well, what if I know what I want my dot to be, but I don't know how to connect that dot to the rest of the network? So we became interested in this idea of designing biological pathways towards specific chemical targets. And the words on the left just say, this was an interesting molecule to work with. It was very expensive when we started it. There wasn't a known biological route to get towards either the lactone, which is shown in the upper right, or its hydroxy acid analog. And the reason the hydroxy acid analog was significant was that it turns out nature actually deals with these kinds of molecules a lot. And the details of those boxes don't really matter. The point here is that these kinds of structures are known to occur in naturally occurring polymers and in polymers that you can produce by providing precursors to the cell. So George talked about non-biodegradable polymers. So some of these are biodegradable, some of them are not biodegradable, but the focus has largely been on the biodegradable ones, as he pointed out. So we knew that nature could make polymers that were wholly composed of this hydroxybutyrate molecule. We also knew that nature could make polymers that were copolymers of the hydroxybutyrate and the hydroxyvalerate, and that meant if we could figure out how to make the free monomers, not the polymers now, but these individual molecules here, then that would give us perhaps a platform to be able to make other molecules that were structurally related. So actually, apologize, I left the reference off of this. This is from a paper we published in Nature Communications in 2013, I think it was, where the base pathway here is if we just have any carbon source available to the cell that will allow it to have this essential precursor called acetyl-CoA, then we could get hydroxybutyrate to be produced. If we were able to provide precursors that could then be activated to the relevant substrate molecule, we can make a longer chain hydroxyvalerate. Here is the molecule we were targeting initially, which was this lactone, and so we were able to make that by providing a different precursor. We could keep going further and further down the chain length to make hydroxyhexanoic acid in this case, and we could even make a branched molecule here called 4-methyl-3-hydroxyvalerate. And that actually became a precursor for us to think about making a biofuel molecule. And so the focus here was to try, this was actually a project that was funded by one of the oil companies who said, give us something that would be as good as gasoline. And the criteria we were given was that to perform like gasoline, it needed to be a molecule between 5 and 8 carbons long. It could have one oxygen group, and it needed to have a branch point in some way. So if you compare, for example, here versus here, the fully linear aliphatic chain versus the branch chain gives you significantly better properties. So we were able to take this precursor, and then based on what we knew about how cells were able to make butanol, which at the time was very popular as a renewable alternative to gasoline, we could design now a pathway to allow us to make this molecule here called 4-methylpentanol. And what's shown on the right is a comparison for the energy density, which is just a way of saying how much energy can you get per volume of material, so how much bang for your buck there on a volumetric basis. And you can see butanol was a little bit better than ethanol. Ethanol ends up being about 2/3 of what you get for conventional gasoline. Butanol gets you up to about 80%. And the molecule that we made when we did calculations, we had about 97% of the energy density of gasoline. And that extra branch point there gave us a high octane number as well. And so Chris talked about the complex engineering that was done in the chloroplast. This relied upon a lot of genes, but also from different organisms. So we really had to take advantage of our ability to scour through nature and ask, what is nature capable of doing from a synthetic perspective, from a perspective of doing chemistry, and then mix and match different reaction steps together to now mimic a pathway that was known in nature in order to produce something that could not be found in nature. So this, as I pointed out, was something that was of interest to show. They are not funding the work anymore. We had pretty high selectivity. We had really low productivity. So I'm going to come back to that as a challenge, where what we have found is that for the most part, we can make most of the stuff that we set out to make, because we've gotten better at just saying, no, I don't even want to try to make that because that's going to be too hard. So we sort of understand the parameters around what biological systems or microbial systems can do pretty well. But to get that towards numbers where it's really going to make a difference as far as the kinds of impacts that we're talking about continues to be a challenge. One of the difficulties that we had here was that 80% is pretty good for a first proof of concept, but it's still not, again, at those really high selectivities that you would need to be competitive going forward. So we've collaborated with Bruce Tidor at MIT, who's in the computer science and AI lab and also in biological engineering. Bruce is a computational structural biologist. And so the question we had was, we know that there's this multi-step pathway. Without going into too many details, we wanted to focus on one particular reaction step to see if we could alter the selectivity to be able to make more of the specific branch chain molecules we were interested in and less of this undesired byproduct. We ended up, just as an interesting aside for those of you who do more of the microbial engineering bit of this, we actually ended up making the polymers inside those cells and then depolymerizing them as a way of getting a readout of the selectivity, because it turns out that we identified other bottlenecks in our pathway. So that's another challenge in terms of being able to figure out. Chris mentioned codon optimization. We've had that problem where optimized genes are really terrible. And so we actually had a significant analytical burden in trying to bring this project forward. So working with Bruce, what we were able to do was to create a number of rationally designed mutants. And I told you initially the selectivity was about 80% in favor of wild type. So that's consistent with this roughly 3-to-1 preference for the desired versus the undesired. But we got mutants that were 4 to 5 times better than this, so that we could get well above 90% now in terms of the selectivity for the compounds that we were interested in. So the point I wanted to make here is the fact that we actually do have a bit of flexibility in terms of what biology can do. There are companies who have a lot more resources than I do, have been able to design pathways and get them all the way through to commercialization. But if we think about this back from this original idea I gave you, that what we really need to be able to do is to figure out how to make as much as possible without relying upon unsustainable feedstocks. We have to be able to do more than this. And one of the challenges that we have is that biology uses a very small amount of the periodic table. So 4 elements represent more than 90% of the dry cell weight of biological systems. And there's a lot more space on the periodic table to be explored. And so some of that space is far more easily explored with harsh industrial chemical practices that have lots of emissions, that use a lot of solvents, that actually contribute to emissions, and that are very energy demanding as well. And so I think one of the big challenges to address is how do we actually increase the diversity of what we can make, both in terms of the complexity of those actual molecules, but also in terms of being able to access more of the periodic table. And I think one of the things, even though this is a synthetic biology workshop, one of the things that I'm trying to be more mindful of, and my own background is more in the biological applications, but to think about the fact that there are a lot of people who do chemistry who are also trying to figure out how to do that in as least a damaging way as possible. And in many cases, doing it in a way that they actually can be beneficial in terms of really reducing the footprint of chemical manufacturing. And so one of the things that I'm increasingly interested in is finding opportunities not to just vilify the chemical industry, but to figure out how can we work together in these hybrid systems to be able to get high efficiency, low environmental burden, but still be able to give humans access to the various materials that we need. So if we go back to our generic system here, I mentioned feedstocks as one of the other things to look at. I think we can all agree, and there's certainly a consensus that you don't. I always love this picture of just putting an ear of corn in the trunk of your car. You want to go away from that. And what's interesting is I think historically over the past 10 years. The debate was really on food versus fuel. But Chris made a very good point, which is that agriculture is very energy demanding as well. If you look at the burden associated with with ammonia production and fertilization, there were a lot of studies that were done that showed if you actually used corn as the input for bioethanol, for example, that it actually didn't give you a net reduction in greenhouse gas emissions because of all the things that Chris talked about and the burden associated with not just manufacturing, but transportation in particular. So, we know that that's bad. There has certainly been a lot of interest in using alternative feedstocks, including specific feedstocks that can be used for chemical manufacturing. There's progress that's been made there, but it hasn't been fast enough. There are not enough systems or enough commercial operations in existence where you can really take advantage of what really would be dedicated crops that have a low demand for fertilization, a low demand for water, to be able to convert them to things that are of interest. I think one of the very interesting areas is with municipal solid waste, food waste. The numbers are always very interesting, and I apologize, I don't remember them exactly, but certainly within the developed world, there's a food distribution problem, much more than a food availability problem, and as en route to solving that, what we end up with is a lot of waste. And so the question is, can you actually take advantage of some of that waste to be able to convert it into something that's more useful? There are also non-edible wastes that come along with that as well. So you can see banana peels in that picture, things that we may be able to take a little bit better advantage of. And to George's point, in some cases, being able to convert that into something that's more that has better sequestration properties than just actually allowing it to be decomposed into methane and CO2 all over again. I don't see him here, but I saw on the invitation list Michael Kupke from LanzaTech. LanzaTech is a company that's looking at using gaseous feedstocks as input to be able to produce, I think their first products is actually making bioethanol, but essentially taking the waste material from steel plants are the first demonstration and commercial facilities that they have, and being able to now sequester that gaseous material into chemical form that can then be used for something else. And George talked about non-degradable plastics or non-degradable polymers. Those are great as long as they don't result in this, right? So the problem right now is that we have a large amount of non-degradable polymers that are causing tremendous environmental damage. And so there's a lot of excitement in the isolation of organisms that were able to grow on polyethylene terephthalate or PET plastic as a carbon source, but there certainly is a lot of work that has to be done there. And then I want to point out, when we talk about CO2 as a feedstock, George talked a lot about photosynthesis and the natural ability of the environment to be able to take CO2 out of the air. I will say, in my experience, and we don't work on photosynthetic organisms in my lab, or from what I read and from the people that I talk to, we still don't quite have the rates that we need to make a significant impact on looking at engineered biological systems for true CO2 fixation. And that's true whether or not, certainly if you're talking about ambient air, it's a real problem. But even if you have the ability to have concentrated CO2, there's been a lot of companies that have come and gone looking at trying to have photosynthetic production of fuels and chemicals. And a lot of the problems, I will point out, I think are engineering problems rather than scientific problems. They are problems with light penetration in cloudy systems and in dewatering and the energy associated with all of that. So I think what I loved about George's talk as a prelude to this is I think what's clear is that there is tremendous potential for biology that's naturally occurring to be exploited, to be engineered now to supercharge its ability, our ability now as scientists and engineers to really greatly accelerate the extent to which we can remove CO2 from the atmosphere. But I also think it's clear that there's a lot of work that needs to be done. We're not at the point now where we know what the solutions are, and it's just a matter of having the right magic or whatever it is, the right person that's leading the charge. There's still a lot of technology development that has to happen. So just very briefly, I want to talk a little bit about process control in cellular factories and how we can think about the advantages that can come from that. The joke I always make here is that I was an undergraduate at MIT. I think we are the only chemical engineering department in the country, actually in the world, that does not require process control. So I have no idea what this picture means. But I know that it's a process control diagram from chemical engineering. And so the question here is how do we actually apply some of these control systems into cellular factories? So a different molecule that we've worked on for a long time is glucuric acid. The pathway is shown there. I just want to highlight and draw your attention to this intermediate glucose 6-phosphate, which is the first intermediate along our desired pathway, but it's also an essential intermediate in cellular metabolism. And so the challenge that we have with something like this is that the cells would rather not make our product. What they would prefer to do is to be able to take that carbon source and use it for its own purposes. So we've worked for a long time on systems to be able to dynamically regulate metabolism so that we allow cells to first use carbon sources for growth and then shut that ability off or tune it down significantly and allow the precursors to be used now for product formation. So we can do a little bit of genetic engineering to be able to restrict the options that we have for where that glucose material can go. And then we apply what we call metabolite valves to be able to regulate a key enzyme in the pathway where initially we allow the cells to use carbon source for growth and for energy. And then if we throttle that valve a little bit, we expect for it to allow us to have a buildup of the substrate to go into a product of interest. So we've worked on this for a long time. We've done this in a number of different ways. In our most recent work, we've actually used quorum sensing as the triggering mechanism to be able to have this shift takes place. And again, the purpose for us in doing that is that it allows us to have something that may be more viable for large-scale production. So the interventions by humans is minimized in that case. There's, again, a bunch of words on this slide. I'll just say we want to be able to do our development at small scales, but we need to be able to have some confidence that it's going to work at larger scales. So we can test this across these various scales. Our base for doing this are 250 ml shake flasks, so about 50 mils working volume. In this particular case, what we found is that our baseline strain wouldn't make any product at all. That's the blue bars. And instead it just made all byproduct, which is acetate. But we could identify a strain that switched at just the right time to give us all product and no byproduct. And then if we looked at a smaller scale down model, it actually mimicked very well what happened in the shake flask. And so we do a lot of our work now at the smaller scale to be able to have the kinds of throughputs that Chris Voigt was talking about. But then we want to also be sure that this will work at larger scale as well. And so we did show that when we went into a controlled environment, this is a closed environment, we have less control. When we could control pH, then we did suddenly have now our wild type strain being able to make product, but it made much more of itself, so more biomass than product. And our strains that were the best performing strains continued to be so. So we did see trends that were consistent across scales, and we actually were able to, with this very precise control, actually able to produce product where we couldn't in a system where the cells were just allowed to grow freely. So this work, again, was published a couple of years ago. We have work that is actually just being published this week where we've shown that we could actually take two different control points to be able to show, again, the ability to more tightly regulate metabolism. So in this case, we've got now a quorum sensing molecule that will control one of our two systems that's called the Lux system, so that as we increase concentration here, we're able to turn on gene expression there. Excuse me, and that's shown here, where if you wait over time, you can see gene expression being turned on. We had a second system that actually uses the same quorum sensing molecule, but it has a different regulator. But it behaves very similarly in the sense that you have gene expression turned on as a function of time. And I mentioned the fact that we could find the right time for this switching to happen, and that's actually controlled by the rate at which you produce the signaling molecule that's used here. And so as you increase now the expression of that gene, then you're able to increase or decrease the timing at which the switching occurs. And then we have another control point because one of our two circuits is only affected by this particular regulatory molecule. And so we can actually alter that by modulating its expression levels. So we can demonstrate that this works by using two reporter molecules. This is a red fluorescent protein and a green fluorescent protein. And what you can see is that in the case of now looking at the one on the right, that this is a factor of the ESA-I level, so what our signaling molecule is, but it's independent of what this regulatory concentration is. But the other circuit is dependent upon both of those. So that actually gives us two different knobs that we can turn to be able to modulate activity. I'll briefly introduce a new pathway here where the design objectives are to be able to delay expression of these two upstream enzymes because we know that this intermediate is inhibitory. So we want these two enzymes to be produced first so that there's plenty of them available before the cells start making an intermediate that it can't use very well. And then one of the substrates that's required is this compound called malonyl-CoA. And malonyl-CoA is essential for the cells to grow. So at a certain point, we also then want to be able to tell the cells to stop using malonyl-CoA for growth, and that will allow it now to shuttle more of that material into our pathway. So we're going to, in this case now, independently vary two different control points to be able to look for the best performance. So we used one of our regulatory networks to be able to control these two upstream genes going from on to off. What you can see is that if we don't turn them on at all, the activity, the productivity is very low. If we turn them on but turn them on too early, it's also low. And so if we find just the right time of turning on those genes, either by adding this molecule or searching through this library to identify a cell that knows when to trigger at the right time, then you can see we do substantially better in the productivity. We then have a separate mechanism using some of the tools that George talked about to control the enzymes that will consume malonyl-CoA. And if we actually have both of these combined, then we get substantially improved production. So we end up with the two of these together about 2.5 times more than using either of them individually. So let me summarize for the work we do in our lab. We think biological synthesis is an effective way to be able to produce chemical compounds, again, trying to displace the feedstocks here. We've been able to develop additional methods that go beyond what the traditional methods are for medical engineering, and those include protein engineering as well as dynamic control of gene expression. And I'll just summarize this last part if we go back to the diagram that I gave you by saying I think we need to be concerned both about what we can make with biology, being able to expand the feedstocks, and that definitely includes being able to increase the rate at which we can take up CO2, and then being able to have as tightly controlled and refined systems as possible to be able to increase the capacity of mitigating greenhouse gas emissions. So I'll end just by thanking the folks who've done the work and all the various funding sources over the years, and I'm happy to take a few questions before the break.
Morning. I'm Todd Kukin from NC State in the Genetic Engineering and Society Center. I thought it was interesting that I think I heard both you and Chris say that when you started this work, you weren't thinking of it from sort of a climate change impact ability.
Yeah.
But clearly the work you all are doing actually has those potential impacts. So I have kind of two questions, which is how can those of us sort of in the environmental space better interact with groups like yours to get you, if you're not currently doing it, to think more about how you can address some of those issues. But at the same time, I'm curious, like, if you were thinking of climate change when you started this, would any of your research have kind of gone in different directions? Because I also wouldn't want to kind of mess up or
Yeah.
mess you guys up in terms
Yeah.
of like getting any of those benefits
Yeah.
out?
So I believe in being very honest and answering my questions, right? What, I'm not saying other people aren't, but usually I end up telling truths that people are surprised to hear, right? So when I first started this, I was first starting my lab at MIT, and my concerns were less lofty. Right? So it was, okay, what are some interesting problems that seem to be tractable enough that I can get tenure? Right? But so the context as well for when I came into the work there wasn't constant talk. There wasn't the constant drumming of climate change. I actually came to MIT from a pharmaceutical company. I think if I had not done that, then perhaps if I had been in an academic environment, then the exposure might have been greater. So to get to a little bit of your point, which is how do you have more of that focus? When I started at MIT, it's a very interdisciplinary place, and so the exposure was much greater in terms of the impact of the work that we do beyond where I initially was thinking about it, right? So I think the short answer to how do we do that is that, at least from my perspective at the university level, I think universities are well designed to do that. The exposure that students and postdocs have in this very multidisciplinary environment, even the people that they come across having lunch, is very different than if you're in more of the corporate-centered environment that I came out of. So I find for my current students who are coming out, both because of the combination of them coming from an academic environment and because of what today looks like, they are much, much more concerned about these issues than I was. And so it was naive, it was selfish, right? And I think that's why I didn't think about it in that way. To your second question, in terms of would I have done things differently, honestly, I don't think so. And part of that, to be blunt, has to do with tenure as well, right? So the example I'll give you is, for years, I would get asked questions, why are you trying to make acids in E. coli, right? Why don't you go find some acid-resistant, wonderful organism that you can dig out from some waste facility that deals with acid? You'll find things that are acid tolerant there. And the standard response I had was that E. coli was the best organism to work with on the time scale of someone who was trying to get tenure. Right? And that was very true. And in fact, when early in my career, within the first year, I talked to a senior colleague and said, I don't think we have access to the right organisms. I think there are bigger problems that we have to solve, and we're going to need to be able to domesticate more of these organisms. And the advice I was given was, don't do it, because you're not going to make sufficient progress in time to be able to attack that. So we're now looking at other kinds of organisms where those pressures aren't there. And I actually think that is, again, these are the hard truth. I think that's a legitimate issue with young academics, which is that there has to be a balance between picking problems that are impactful and picking problems that are tractable. And those aren't always the same. I feel like I need to have a happier answer. Yeah, June.
So, Chris, now that you are a tenured MIT professor, we can be more...
Now I'm just tired, June.
Okay, so now we can be more creative. Yeah. And one of the things that always occurs to me every time I hear you talk is plants secrete, and some of them secrete ridiculous amounts of goo. So why not engineer that? That's a somewhat carbon negative system, and then just take them and kind of rinse them in whatever you want, collect the goo, and go from there.
Yeah, I don't know anything about plants. That's the answer to that. I also, you've heard me say this, I also say I think you can judge the patience of individuals by the growth rate of the organisms that they work with. I'm not Vibrio yet, but I'm E. coli, which isn't that far away. And so, yeah, I mean, I have a lot of admiration for Chris for going into plants so much, having come from a strictly microbial background. I need to take some bravery pills. I don't have the stomach for it.